Hello, my name is Professor Hudson. Let's continue with the narrated PowerPoint lecture for Chapter 5, Section 5.2. Section 5.2 covers how do substances move across membranes. This slide shows a real-life example of osmosis and tonicity at work with respect to red blood cells. Red blood cells have an internal concentration typically around 0.9%. Red blood cells put in a isotonic solution that are also at 0.9% are content. The movement of water in and out of this cell will be roughly equivalent because the concentration of the solution is at 0.9% and the concentration inside this red blood cell is at 0.9%. We say this solution is isotonic to the red blood cell. When red blood cells are put in a hypertonic solution, or one that is more concentrated, say for example, if red blood cells were put into a hypertonic solution of say 1% or 10%, in this situation, the cells will shrivel. The higher concentration of solute outside the cell means that there is less water present. In this situation, water will move from inside the red blood cell, where it's higher, to outside the cell where it's lower. This is called crenation, when an animal cell such as a red blood cell shrivels. When red blood cells are placed in a hypotonic solution, a solution that is less concentrated than the inside of the cell, say a solution that was 0.5% compared to the inside of the red blood cell, which is 0.9%. In this situation, the hypotonic solution has less concentration of solute and more water. In this case, water will move from outside of the cell into the cell, causing it to swell. Eventually, the red blood cell will burst. We call this lysis. When cells are placed in a hypertonic solution, such as shown here, they shrivel owing to water loss. When animal cells are placed in a hypotonic solution, they swell owing to water entry. Cells in an isotonic solution remain unaffected. These same principles happen with plant cells, except plant cells have a cell wall that can keep them from bursting and can also make it less noticeable when they shrink up. Let's take a look at an animation that is supposed to illustrate what happens to red blood cells in hypertonic and hypotonic and isotonic solution.
The image on the left is human erythrocytes, or red blood cells. The image on the right is enhanced with filters. These living cells are in isotonic solution. Note the lozenge shape. Now the solution has been changed to have a high salt concentration. It is hypertonic. The red blood cells shrivel and become crenated. Finally, the solution has been changed to plain water. The cells swell and burst, leaving empty membranes behind. This phenomenon is called hemolysis.
This slide shows the effects of osmosis and tonicity on plant cells. Osmosis and turgor pressure affect how water moves in a plant. Water flows into a plant cytoplasm because it is more concentrated than the extracellular fluid. This would be the equivalent of placing the plant cell in a hypotonic solution. Water flows into the vacuole because its contents are more concentrated than the cytoplasm. Water pressure within the vacuole causes turgor pressure. The cell wall keeps the cell from bursting as the water flows in, creating pressure. In the opposite scenario, if the plant cell is in an environment where it's hypertonic and there's less water outside of the cells, then water will actually move out of the cells into the surrounding environment. And we call this plasmolysis. Plant cells in a hypertonic environment will shrivel, however, it is not as noticeable because of the cell wall. However, when plants look wilted, that is evidence of plasmolysis, or water leaving the plant cell. Let's review. Question 14. If the inside of a cell is 1% sodium chloride, which solution is isotonic to the cell? Choose the correct answer. In this question, you are choosing the solution that is isotonic to the one in the question. Remember, isotonic means the same concentration. Question 15. When a plant cell is placed in a hypotonic solution, which of the following will occur? Choose the correct answer. This slide is illustrating some differences in transport, particularly active transport. During active transport, membrane proteins use cellular energy to move molecules or ions across a plasma membrane against their concentration gradients. With passive transport, such as simple diffusion, facilitated diffusion, and osmosis, the molecules are moving from high to low concentration. In active transport, or energy requiring transport, the molecules are moving from low to high concentration. Often this requires pumps or channels or carriers to help the molecules move against the way they normally would move. Let's look at an animation of how the sodium-potassium pump works. This is an example of active transport.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. 1 phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
Let us review. Which of the following correctly describes active transport? Question 16. Choose the best answer. Let's finish up this section by looking at some other methods of active transport. In this figure, you are looking at something called pinocytosis. Pinocytosis is when the cell engulfs something small using the cell membrane. This energy requiring process is called endocytosis. Pinocytosis is sometimes called cell drinking, where the cell is literally sipping liquids in the environment. Another form of active transport, or transport that requires energy, would be receptor mediated endocytosis. This is when the cell is swallowing something specific by binding receptors in the membrane. This is a form of endocytosis. And then another mechanism of endocytosis is referred to as phagocytosis. In phagocytosis, the cell is eating large molecules or particles. This is in contrast to pinocytosis, when the cell is drinking or taking in liquid. This is a form of endocytosis. So there are three forms of endocytosis. There's pinocytosis, receptor-mediated endocytosis, and phagocytosis. When the cell needs to get rid of something, it can use the membrane and ATP to move materials out of the cell. We call this process exocytosis. Vesicles containing the material to be expelled move to the cell surface, where they fuse with the cell membrane, allowing their contents to diffuse into the outside fluid. This is exocytosis. Endocytosis is when the cell takes something in, and exocytosis is when the cell secretes or excretes something into the environment. Let us look at an animation of endocytosis and exocytosis.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use three basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
Now that we have talked about how substances move across the membrane, we will finish up Chapter 5 by talking about how do specialized junctions allow cells to connect and communicate in the next set of narrated lectures.